Welcome back. We are going to finish up the endocrine system by talking about some of the chemical properties of hormones. So we have two classes of hormones. We have what are called steroid hormones, which are lipid-based, because remember that steroids are one of our types of lipids. And we have non-steroid hormones, which are amino acid-based. So here we see steroid. Remember that all steroids have this four-ring structure. So you can see here there are some pretty minor differences between testosterone and estrogen, and yet pretty major effects that are pretty different. Monoamines are made from a single amino acid. They retain the amino group, so right here and there. And then peptides are going to be multiple amino acids strung together. So the control of release is something we need to look at here, because there are three different ways to control the release of a hormone. We've basically talked about them in the previous lectures without really getting into it. Here we're going to look very closely at them. So the one that we've probably talked about most is this one, the hormonal stimulus. This is where the hypothalamus has a releasing hormone that triggers the anterior pituitary to release a stimulating hormone, which causes the peripheral gland to release a hormone. So what this is a hierarchy of hormonal control. A hormone causes the release of another hormone, which causes the release of another hormone. This has to do, the neural stimulus has to do with the adrenal medulla. So this is your sympathetic nervous response. So this is direct neural innervation to the gland, causing the release of epinephrine. The only gland that uses this is the adrenal medulla. And then the third is what's called humoral. So not hormonal, but humoral. Humors mean fluids, usually blood. So this is a release based on blood levels of something. So calcitonin, parathyroid hormone, insulin, and glucagon, those all function on the level of something in your blood. So here, parathyroid hormone is going to be released simply when there is not enough blood calcium. And when there's too much blood calcium, calcitonin will be released. Insulin is released when blood glucose goes above 90. Glucagon is released when it drops below. So these don't necessarily have anything telling them to do anything other than working off of the homeostatic level. And then they're going to work on negative feedback loops. So you've got multiple different ways to control hormone release. Now, sometimes this works really, really well. Actually, most of the time it does. But I'll give you an example of a time when this doesn't work great that's going to fall into this hormonal stimulus here. So let's talk about goiters. What a goiter is a grossly enlarged thyroid gland. Okay, so this is actually his thyroid gland that has enlarged. It's undergone hypertrophy due to an increased workload. And this is where this concept of a breakdown in negative feedback comes in. So what's supposed to happen is the hypothalamus releases thyrotropin releasing hormone, which targets the anterior pituitary, which releases thyroid stimulating hormone, which targets the thyroid gland, which releases thyroid hormone, which causes thyroid hormone levels in the blood to increase, which should then come back up and inhibit the anterior pituitary and the hypothalamus. But what if your thyroid gland can't make thyroid hormone? The most common cause of a goiter is simply a lack of iodine. T4 requires iodine. If you don't have enough iodine, you can't make T4. If you can't make T4, you never release it, which means that your blood level never goes up and you don't have your negative feedback. So what happens is this is always going. The hypothalamus is always telling the anterior pituitary to tell the thyroid gland to make thyroid hormone. So the thyroid stimulating hormone is always being secreted, and the receptors on the thyroid are consistently being activated, and the thyroid tries very hard to respond. And in trying, the cells get bigger and bigger and bigger and bigger. They're undergoing hypertrophy because their workload is constant. Iodine supplements can eliminate a lot of goiters. We don't see a lot of goiters in the United States because we have iodized salt. At some point, we started putting iodine in our table salt, and that gave us a source of it. Now, depending on where you are, a lot of the stuff that's grown in the soil will either contain iodine or it won't, depending on where it's at. So just an example of how sometimes these mechanisms of control don't work all that well. All right, so let's talk about the mechanisms of action. So steroid hormones are going to act on intracellular receptors. A steroid hormone is a lipid. It's going to be able to move directly into the cell, and it's going to interact with nuclear receptors. It's going to turn on gene transcription, which is going to then eventually have whatever the effect is going to be. So these tend to be a little slower because we actually have to activate transcription and translation. Non-steroids can't enter the cell. So their chemical property means that they are not capable of diffusing through the lipid bilayer, which means that they are going to have to activate a membrane-bound receptor and then use what's called a second messenger system. And often these are linked to what are called G proteins. The G protein activates another enzyme that gives us a second messenger. In this case, the example is cyclic AMP, which then turns on an enzyme, which turns on another enzyme, which turns on something, and eventually we get to our effect. So the cyclic AMP is a very common one. All of these different types of hormones use it. The PIP calcium is also another very common one. But basically, this goes down to their chemical properties. A steroid hormone can diffuse through the lipid bilayer, whereas a non-steroid hormone cannot and will have to use a second messenger. So then the question is, they've activated their receptor, what do they do? There's multiple things that a hormone could do. It could change membrane permeability, which would change membrane potential. Okay, so that's usually going to deal with opening ion channels or closing them. Cause the stimulus of, or stimulating synthesis of proteins or something that's going to be a transcription regulator or something like that. Turn on or off enzyme systems. Cause a secretion of something else. Stimulate cell growth. So there's multiple different possible actions that can occur. Hormones are released into the blood, which means that essentially every cell in your body is going to be exposed to every hormone you have, and yet only certain cells react to the hormone. Those are called your target cells. They react because they have a receptor for the hormone. Not every cell has a receptor for every hormone, and if you don't have the receptor, the hormone has no effect on the cell. So some target cells are highly specific. So adrenocorticotropic hormone, the only cells in the whole body that have a receptor for that are on the adrenal cortex. That's it, causing the release of cortisol and aldosterone. So that's it. Those are the only things we have. Whereas thyroid hormone receptors are found on almost every cell in the body. So you're going to have a really widespread effect in terms of that. Now, the activation of the target cell is going to depend on a few things. First of all, the blood level of the hormone. Are you secreting enough? Are you secreting too much? What's going on there? The number of receptors is going to come into play. So if a target cell upregulates, it's going to increase the number of receptors in the membrane. It's going to increase its sensitivity to the hormone, and you're going to get a stronger response. It can also downregulate, so it can pull the receptors out of the membrane, which would mean it will be less sensitive, and you will get a much smaller response from the target cell. Remember, cells are dynamic. They can absolutely take things out of the membrane, put them back in, take them out, put them back in. This is upregulation and downregulation are very common. And then we have the affinity of the receptor and its hormone. Is this a really strong interaction where that receptor grabs a hold of the hormone and never lets go? Or is it coming into the receptor, kind of activating and bouncing back out? The stronger the affinity, the stronger the activation. Now, hormones often have interactions with other hormones. So one type of interaction is what's called a permissiveness, where one hormone can't really work without another one being present. So growth hormone and thyroid hormone fall into this. So growth hormone can't really work if thyroid hormone is not there. Another example is estrogens and progesterone. During the early portion of a menstrual cycle, there is high estrogen, and that estrogen causes upregulation of progesterone receptors so that when the progesterone shows up, it can actually work. Without the estrogen driving the increase in receptors, then progesterone isn't going to be able to work. You can secrete it, but there wouldn't be any receptors. So there's a permissiveness interaction. Synergism is when two hormones work together on the same concept. So FSH and testosterone work together for sperm production. Neither one of them can create a whole lot of sperm without each other. Okay, they both have to be present at the same time. When you get them both there, you get massive sperm production. We're talking somewhere in the neighborhood of like 300,000 per minute when this is happening. So that's a synergistic effect. Epinephrine and glucagon have a synergistic effect. When you are releasing both of them, they cause even more glucose to be released into the blood. And then antagonism is when you have an opposing action. So calcitonin and parathyroid hormone oppose each other. One is going to cause you to pull calcium out of the blood. One is going to cause you to release calcium into the blood. Insulin and glucagon have opposing effects. One causes the release of glucose, one causes the removal of glucose. So when you think about all of these hormones in the blood together, it's not surprising that they do interact with each other.